Good morning, everyone. Thank you all for coming, joining us here today. Our today's seminar is going to be given by Brent Nannenga. He's going to tell us about microcrystal electron diffraction, MicroID, methods and applications. And before he gets started, let me just give you a brief overview on Brent Nannenga. So Brent Nannenga is an assistant professor of chemical engineering in the School of Engineering Matter, Transport and Energy at Arizona State University, and a member of the Center for Applied Structural Discovery at the Biodesign Institute. He received his PhD in chemical engineering from the University of Washington in 2011. Following his graduate studies, he did his postdoctoral research at Janelia Research Campus in the lab of Tamir Gonen, where he focused on the development and application of cryo-electron microscopy, specifically the development of microcrystal electron diffraction, MicroID. He joined Arizona State University in 2015. His research has been focused on developing and using methods for high-resolution structural determination and employing structural insights gained from these methods in order to engineer biomolecules and materials with novel and unique properties. His work has been recognized by awards, including an NSF Career Award, an Air Force Office of Scientific Research Young Investigator Award, and the 2020 Barton Medal from the Microscopy Society of America. And with that said, Brent, whenever you are ready, the stage is yours. Welcome.
Great. Thank you, Jose. So, and thank you all for coming. Thanks for inviting me here. I'm very excited to talk to you all and to visit your campus, and especially excited to reconnect with Jose. We were both at ASU at the same time, and I miss being able to go down the hall and talk to him about various crystallography questions and cool things. So thanks for the invitation. So as Jose mentioned, I'm going to talk today about this technique of microcrystal electron diffraction, or MicroID. I'm going to the first bit of the talk will be focused on what is the technique and how do we use it, and how do we, what are the steps and what's the workflow of the technique, and then I'll move into applications. How do we use it? And it's going to mostly focus on biological samples, and then towards the end I'll give some examples of small molecules, because that's kind of been I don't want to say unexpected, but kind of a surprising application of MicroID that's kind of exploding right now is applying this to small molecules and samples from material science. When I started my postdoc, we were focused mostly on proteins because we were protein crystallographers, and now I think when I do MicroID, probably 90% of the time I'm actually doing small molecules now as opposed to proteins. So I've had to learn, how to do non-chiral space groups that I had no idea what those even were as a protein crystallographer. So now I have to know all sort of 230 space groups, but it's been fun. So as Jose mentioned, in my lab, we have a lot of different projects. I'm going to mostly talk about like I said, the methods and applications for cryo-EM, of which MicroID is one of these methods. And so just to orient everybody is probably familiar with cryo-EM. It's a very popular technique right now. And when people think of CryoTEM, they normally think about single particle CryoTEM, right? That's where you image individual molecules, you average those together, you solve three-dimensional structures from those images. But there's a lot of different techniques within the broader field of CryoTEM. For example, imaging-based techniques, again, like most people know single particle. But there's also electron tomography where you look at whole cells. And then you also have crystal-based techniques. So one of the first actual applications of cryo-EM was actually using two-dimensional crystals. So a lot of you might be familiar with Richard Henderson, who won the Nobel Prize for single-particle cryo-EM a few years back. Actually, a lot of his work in the 70s was actually on two-dimensional crystals, doing diffraction and imaging of crystals. That's where they found the first organization of alpha helices in the membrane on Bacteroidopsin back in the 70s. And then more recently, using electron diffraction of thin three-dimensional crystals, and that was a technique we developed called MicroID. Instead of using thin two-dimensional crystals, saying how would you collect data if they're not two-dimensional anymore but three-dimensional. There were some technical challenges to overcome in order to study those kind of samples, and we'll talk about that here in a little bit. So first off, I mean, I probably don't need to talk a lot about this to this kind of audience, but I like to first start, well, why do we need a new method for crystallography? What's the problem? You know, X-ray crystallography is fantastic, right? I mean, it solved tens of thousands, hundreds of thousands of structures. So what's the problem? Well, when you have crystals that look like this, right, there really isn't a problem, right? You take these big, beautiful crystals, the synchrotron, they diffract to 1.5 angstroms, you solve your structure, and you go home. But the problem is that's not very common. Usually you get something like this, and the protein crystallographers in the room recognize this, right? You get a bunch of junk, maybe some tiny little crystals, and then maybe you spend the next 3 or 4 years trying to make them bigger, and maybe they never get bigger, right? So there's a need in crystallography to be able to study smaller and smaller crystals. And something we didn't appreciate too is this need is also exists in the small molecule community as well. We were a bit naive and thought small molecule crystallographers, oh, you can always grow big crystals of those, but that's actually not the case either. And so this also holds true for small molecule material science as well. There's a lot of techniques. Synchrotrons are getting better. They're shooting smaller and smaller crystals. You've got free electron lasers that are able to shoot smaller and smaller crystals. But we took a different approach and said, well, electrons interact with matter a lot stronger than X-rays. So you could use electrons could be used to collect diffraction data from very, very small crystals. So that was the approach that we took. Now I just want to bring this point up too. This gets lost a lot of times for people who aren't familiar with the technique, and that is the machine that we use essentially is the same machine you'd use for single particle cryo-EM. It's nowadays they're coming up with new diffraction-specific electron microscopes, but for the most part, we use electron microscopes that are also used for imaging. For example, on the Thermo Fisher microscopes or the FEI microscopes that we use, there's a button on the panel that says diffraction, and you push that, and it puts the projection lens system and all the lenses into the right mode, and now you basically move, rather than the image plane hitting the detector down here, you move the reciprocal space, the diffraction pattern to the detector. Same instrument. You swap back and forth between imaging and diffraction. That's a really powerful component of this that I think gets overlooked, is that you have these tiny little crystals, and you can go into imaging mode of the microscope, find where your crystals are, focus on a particular crystal, switch it to diffraction mode, and collect diffraction data from that one spot. Whereas when you're doing X-rays on small crystals, you're kind of shooting blindly a little bit. So that's important to know that we use the same instrument for imaging and diffraction. And then there's some subtle differences in things you need, and we can talk about that in the questions about what would you need to do imaging and diffraction on the same microscope. But in theory, the same machine can do both. Now, the differences between electrons and X-rays, and this is important, again, for the X-ray crystallographers to kind of get a feel for what are some of the differences, important differences. Because really, it's diffraction. So it obeys Bragg's law. It's just you're using electrons instead of X-rays. Well, what does that mean in practice? So I have two pictures here. One, an X-ray diffraction of a lysozyme crystal, and then an electron diffraction pattern of a lysozyme crystal. These aren't the same crystals, right? Because obviously the X-ray one is, a couple hundred microns in size, and the electron diffraction one is a couple hundred nanometers in size. But they're the same unit cell, the same protein. And you'll see the diffraction patterns look different. And that's because if you know, I've heard of what's called the Ewald sphere, right? It's a geometric representation of when Bragg's law is satisfied. So in this cartoon up here, and this is a very simplistic view, but hopefully it gets the point across. In this cartoon here, these spots would be your spots in reciprocal space. So you've got, say, L going up and down, H maybe going left and right, and maybe K is coming in and out of the board, for example. And then the blue line would be Ewald sphere, which is the 1 over the wavelength of your incoming radiation, right? So you've got what's the Ewald sphere here in X-ray. Sorry, there you go. It's curved, and so that's why you get these circles. So one diffraction pattern, you get information about H, K, and L, sort of three-dimensional information from a single diffraction pattern. If you look at X-rays, the relativistic wavelength of an electron is 40, 50, 60 times smaller than what you'd have for an X-ray, which means when you do the inverse of that, the radius of the Ewald sphere is very, very large. Not flat, but almost flat. So an electron diffraction pattern, rather than being sort of curved and getting this sort of these circles, you get just sort of flat planes through reciprocal space, which means you get a lot of information, for example, about H and K, but nothing about L from a single diffraction pattern. And this means that your crystal orientation can't be defined from a single electron diffraction pattern. Now, obviously, if you know your unit cell ahead of time, you could sort of, put that in and predict it. But if you had a novel sample, you didn't know what your unit cell was, you couldn't index this like you do in X-ray, right? For those of you familiar with X-ray crystallography, you take a single pattern, and usually you use more than one, but you can, get your crystal orientation and your unit cell primers just from that first shot. Electrons, you can't. And so this was sort of the first bottleneck that we had to overcome when we were developing this technique. So it's like, well, how does this work? And when people were trying this in the past, what they would do is they would do what you would do for 2D crystals, which is you hit the crystal with so much dose that you get one shot and that's it. Well, if you can't index off of a single shot, that's a problem. So our thought was, this other slide, I'd just like to zoom in here for the crystallographers. While this is not necessarily useless for indexing, it's kind of cool because you can actually see the symmetry. So this one, we actually caught the 001 plane here. And so you can see, you can see your two axes here. You can see the systematic absences showing you've got this screw X here. So it's pretty, but again, kind of useless if you don't know what your unit cell is. There's the systematic absences. So the thought was, okay, well, what if we just back the dose off? So because we have a 3D crystal instead of a two-dimensional crystal, we have many more unit cells, so we could get more signals. So we maybe don't need to put as much dose into the crystal to get a diffraction pattern. So what if we back off the dose? We're still going to have this limit. We just say roughly, let's say, 5 electrons per angstrom squared of total dose that this crystal can handle. And normally for 2D crystals, you deliver that all in one shot. We said, well, what if we back that off way, way back, and then we can take multiple diffraction patterns of this crystal as we rotate the crystal. And then we can kind of reconstruct 3-dimensional reciprocal space and hopefully get the unit cell from that. So that's what we did. And this is one of the first experiments we did on this project in my postdoc. We cut the dose all the way down to 0.05 electrons per angstrom squared. And we could see diffraction that goes beyond 2 angstroms. So that said, okay, we can shoot this crystal 20, 30, 100 times potentially and still get good signal. So that set up then the data collection strategy, which is, okay, we take our crystal, and in this little cartoon here, I'll have a better cartoon that Thermo Fisher actually made, but this is better for demonstrating. You've got the electron source up here, you've got your crystal on a grid on the stage right here, and here's your detector. And then this is again, the cartoon of reciprocal space that you're going to sample. Now what you do is you tilt the crystal, and if you collect the diffraction pattern from this orientation, you would sample that yellow line of reciprocal space. But what we do is we take the stage and we get the stage rotating, kind of like what you do, for X-ray, you'd be oscillating. We don't oscillate because the stage can't quite do that, but we just continuously rotate. And as it continuously rotates, you turn the beam on, and the camera runs very, very fast and records the diffraction patterns as the crystal's rotating, which means you sample this volume of reciprocal space up here. And now you know how each image in your diffraction pattern, which would look like this, you'd have a diffraction movie that would look like this. And you know how many, the angular range of each frame. So you now can sort of recalculate what the 3-dimensional vectors in your reciprocal space, and you can index your crystal this way. So that was kind of the key to making MicroID work, was this ability to shoot a crystal multiple times and then rotate it at a constant rate so you knew how each frame was relative to all the other frames in the data set. So the general workflow for MicroID is, first you obviously have to identify a microcrystal, and we could spend an hour talking about how you do that or what that means, but the point is you find your microcrystals, you put them on a TEM grid, looks like this. You see these little crystals kind of in very low magnification mode. You zoom in a bit more to kind of look at the crystals. You identify, hey, that looks like a nice crystal. I'm going to see if it diffracts. You then put your microscope in diffraction mode. You shoot it, and maybe it doesn't look great. Okay, back in imaging mode, you find another crystal, you shoot that one. Eventually, you find one that looks good, and then you collect a full diffraction data set, like I mentioned, as the stage rotates. You take that, you process your data, and at this stage, the data processing, this looks just like X-ray crystallography. We use all the same programs X-ray crystallographers use, and then you refine, build your model using the same tools that X-ray crystallographers use. So this part of the workflow is very familiar to anybody who does cryo-EM, and this part of the workflow is very familiar for anybody who does crystallography. So no matter which side you're coming from, there's going to be part of the workflow that seems very familiar to you. Now here's the fancy video that Thermo Fisher sold or is showing. So you got the grid, you put your crystals on there. For proteins, we do like we do for single particle, and we vitrify them, we plunge freeze them, so we trap them in a frozen hydrated state to keep them happy. For small molecules, you don't necessarily need to do that. But you take that sample and you load it into your TEM. In this case, it's a side entry holder. We also have auto loaders that we use. You tilt the stage, and then you rotate it as you collect data, and you build up this diffraction. All right, I'll just hold this one while we take that. Yeah, so sorry, catalase, in the 30s, they found these little thin plate catalase crystals, very thin plate-like crystals. And then in the 70s, people actually were trying to collect electron diffraction data from these same crystals. Both Dorset and Parsons, they were looking at how you can hydrate proteins and keep them in the microscope. And then you notice here's Unwin and Henderson. Richard Henderson won the Nobel Prize for single particle. He's working with 2D crystals. They also were trying to do this. And it wasn't until I think the mid-80s when they finally grew bigger, thicker crystals for X-ray that actually solved the structure for catalase. So this was kind of like, oh, wouldn't it be funny if we could use MicroID to solve the structure of catalase from these crystals they had way back in the 30s? Yeah, try that. Oh, there, you started. Yeah, great. And the other reason it's a little loud. The other reason we also wanted to go with catalase is because it's rather large. So lysozyme, protease K, they're kind of small. Catalase is a 240 kilodalton tetramer. So the unit cell is pretty big. It's about 70 angstroms by 110 angstroms by like 200 angstrom. So it's a big unit cell. So we thought, well, this would be kind of challenging for data processing. So we went ahead, we put these little, these thin crystals here. You can see here on the TEM, nice thin crystals, same ones that they saw in the 30s and they're looking at in the 70s. And we collected MicroID diffraction data from it, and we were able to solve 3.2 angstrom structure of catalase. Again, mostly just to sort of prove that we could handle something this large. But what's actually really interesting about catalase is catalase has some interesting ligands inside of it. So there's an NADPH binding site in catalase. So this was sort of the first test of, this is actually the second protein we ever did by MicroID, and there was always well, this is mostly model bias, you're using molecular replacement, so it's all model bias, of course it works. So we said, well, I wonder if we could see subtle differences in our structures. So we looked at this NADPH binding site. And this is what the map looks like around that binding site. Again, it's 3.2 angstrom, so it's not crazy resolution, but this is what it looks like in the structure that we have. And so what we did to test, how give a feel for what could we identify if this ligand was bound or if there's any changes with this ligand in the type of data that we have. And so we found there's actually a structure in the PDB of a catalase variant that doesn't have NADPH bound, which is actually really hard because this NADPH sticks really, really hard to catalase. But there is a structure where they crystallize it without the NADPH. And in that case, you can see that this phenylalanine points down, whereas in the NADPH bound structure like we have, it points up. So we phased their data with this NADPH free structure, knowing that NADPH is actually in there and seeing if we could identify these differences. And so if you look at the map, this is a difference map after we phased with the NADPH free structure, and you'll see, for those of you who aren't familiar with this, right, green density means, hey, there's something in your model, there's something in your data that you're not modeling, and red density means you're modeling something there that your data doesn't support. So you can see, there's a red blob density with the phenylalanine pointed down, and there's a green blob pointed up where it should be, and there's a whole bunch of green density here where the NADPH goes. So it does, everything kind of fits, fits like that. So this was, one of the first demonstrations, like, hey, okay, that was cool, we did this on lysozyme, but this might have some relevance. This might, be really interesting. And so this kind of really kickstarted a lot of the work on MicroID, because this was done about 6 months after the first structure of lysozyme. So fast forward a few years, and we said, okay, let's try to tackle something that's much more difficult than just catalase and lysozyme and proteinase K. So we had an interest in membrane protein crystallography. That's my postdoc advisor, Tamir, is a membrane protein biochemist. So that's really what he wants to study. So we talked about this idea, and then in my own lab, I kind of took this idea and pushed it forward. And the idea is to try to apply MicroID to what's called the lipidic cubic phase. So if you're familiar with the lipidic cubic phase, it's this really, really viscous media that has basically a continuously connected three-dimensional membrane environment. And this is really powerful for crystallizing membrane proteins because it allows the membrane proteins to sort of live happily in this bilayer, but still be able to diffuse around and find other proteins to then crystallize with. And so it's really, really been an amazing technique, especially for the study of G protein coupled receptors, which are a very important class of proteins. But it's very difficult because normally when you crystallize things in LCP, you'll get a bunch of tiny little crystals. All these little specks here are actually crystals. So people use, Jose knows, he's involved in a lot of this work, in using the XFEL then to solve G protein coupled receptors in LCP because they're just so tiny. And so we thought, well, maybe MicroID could be applied to GPCR samples in LCP as well. But as I mentioned, LCP has some interesting properties, and it makes they're pretty much the exact opposite properties you'd want for doing electron microscopy. And that is that it's very viscous. So anybody who does cryo-EM knows you put your sample on the grid, and then what do? You blot it away, right? You have to blot most of your liquid away because the electron beam, the benefit of the electron beam is it interacts with matter very strongly. The negative of the electron beam is that it interacts with matter very strongly, which means when your sample gets thick, you can't see through it anymore. So you have to blot away most of your sample. Well, LCP has the consistency of toothpaste. If you squirt it, it just squirts straight up. So that doesn't work for EM very well because you can't blot it away. You can't get it thin enough to actually see through it. The other problem is that this mixture of lipid and buffer has multiple phases. So it's not just a simple matter of, oh, well, let's just keep diluting it. Because as you dilute it, you can move through the phase diagram and actually end up with a phase that either destroys your crystals or ends up being more viscous than what you started with. So this was done in collaboration with Wei Lu, who is a colleague of ours who's sort of an expert in LCP. So we said, okay, let's join forces. Let's figure out how we can manipulate the LCP so we can make it usable for MicroID. So our approach was to figure out how do we manipulate this so we can get the crystals from the LCP onto the grid. We can see through it. We can collect diffraction data from the crystals in the LCP. So the first method we did was try to use different additives to cut the viscosity. So here's an example. This is a filter paper. This is a little blob of LCP. It's hard to tell, but that's a little blob of LCP on the filter paper. It's just sitting on top. It doesn't do anything. You blob it on there and it just sits there. However, if you mix it with something like silicon oil, we wouldn't use this for proteins, but this is just a demonstration. It'll cut the viscosity and then it'll blot away. The second approach is to enzymatically digest the LCP matrix. So add a lipase. It goes in there and it chews up the monoolein, which is the constituent of LCP, and you get a fatty acid and a glycerol layer. And the crystals go to the glycerol layer, which is much less viscous, and you can blot that away. So we screened a bunch of different additives for what you can mix with LCP to get it to be able to blot. And again, these are droplets on filter paper. So you can see the initial LCP droplet looks like it just sits there. It doesn't do anything. It's like toothpaste. If you mix it with things like MPD, PEG 400 though, you can see it starts to cut the viscosity and allows you to blot away a little bit. So we took those along with our second method where we just enzymatically digest the sample. You can see here's some LCP inside of a tube. It's this white stuff. And then after incubating with a lipase, you get these two clear layers. And the crystals would be here in the bottom layer. And after you put that onto the filter paper, it blots away as well. So we used, as our first test case, we used a soluble protein, proteinase K, which you can put into the LCP. So we said, hey, proteinase K isn't a membrane protein, but we're going to use it as sort of a proxy for this process. We put proteinase K into the LCP, and then we said, can we get it back out? Because proteinase K, if you guys are interested in working on MicroID and learning, proteinase K is really the best sample to use for learning MicroID. So we used proteinase K, and you can see both methods here. Even though it's kind of thick and hazy, we were still able to identify the crystals on the grid of proteinase K after they had been in LCP. Those crystals still diffract pretty well, and we were able to solve both structures about 2 angstroms, looking just like what they looked like without LCP. So we thought, great, this approach works. Now let's apply it towards our G protein coupled receptors. So we use this sort of model GPCR A2A adenosine receptor, and we came to a problem, and that was the crystal forms of these crystals. They're not cubic, but they're kind of fat and chunky, which means we could cut the viscosity away and see a crystal, for example, right here. But by the time the crystal got thin enough that we could actually get the electron beam to penetrate, there were so few unit cells that it only diffracted to about 4.5 angstroms. When you got the crystals bigger in this dimension to get more unit cells, it grew too fat this way. And so the beam wouldn't penetrate. So we were kind of stuck there for a little bit. And then this is where we moved over and started collaborating with my old postdoc advisor to couple the techniques we just developed and how do you reduce the viscosity and get the samples on the grid with a new technique for how do you then control the thickness of the crystals on the grid. And so we use a process called cryo-fib milling. So you might be familiar with this if you know cryo-electron tomography. This is what they use to sort of cut cells down and get really nice thin sections in the middle of cells so they can image and do tomography inside cells. We use it on crystals. And so the idea is this is basically, if you're familiar with this kind of equipment, it's a FibSem. So it's an SEM with the focused ion beam inside of it. And so what you do is you put your microcrystal on the grid here, and then you image it in the SEM. You find the microcrystals, and you pick out what you want, and then the ion beam is coming at an angle, and use the ion beam and basically just blasts away any crystal that you don't want. And you take this crystal that maybe is slightly too thick, maybe it's 10 microns, 5 microns, 2 microns, and you cut it down to this little section to where it's only about a couple hundred nanometers thick. And once you have that, you transfer then that milled sample, it's on a grid too, take it out, put it from the cryofib into the TEM, and you collect your microED data on that thin sample, and you get really nice diffraction data. So this is what the process looks like. Here's a lysozyme crystal. You can see it's actually kind of fat. I think it's about 5 microns thick or 10 microns thick. I can't remember exactly. So small, but way too big, way too thick for microED. You take this crystal though, and you fib mill it. You can see you just blast away the top and the bottom, and you just leave this little thin lamella right here that's only like 200, 300 nanometers thick. Then when you look at it from the top, so you can see here from the top right here, this is again looking at it from the top, there was the big chunky parts of the crystal, and then in the middle is this really thin layer. And that's what we're going to look at in the TEM and collect our diffraction data from. So that was already shown to work for, again, proteinase K lysozyme model samples like this. So we said, well, what if we do the same thing for the GPCR samples? Because even a cryofib won't work on LCP. If you just put the LCP right on the grid, and then you look at it in the SEM, it just looks like a mound of nothing. You don't even know where the crystals are and all of this gunk. And then it would take you forever to actually cut through that big fat layer of LCP to find your crystals. So what we did is we took the same process. We used PEG 400 to cut the viscosity so we could put the samples on the grid and kind of get rid of some of that LCP. And then this is a view in the fib. You can then see the outline of the crystals that you want to mill. So here it is, right here. And this is the problem I was talking about. You see the dimensions. This is the perfect size. It's like 15, 20 microns this way, which wouldn't be a problem width-wise for microED, but it's about 2 to 5 microns thick, which is way too thick for us. So we just take this crystal and basically cut through it. Cut a little window here, cut a window underneath, and make a nice thin 200 nanometer section through this. And then we get this beautiful 2.4 angstrom diffraction pattern. Whereas before, remember I said those little tiny ones, by the time we got them thin enough, they only diffracted about 4.5. So now we can actually collect diffraction data. And we were able to use this and solve a 2.8 angstrom structure of this A2A receptor. And it looks just like you'd expect a GPCR to look like. Now the resolution for this is not as good as what was done for the XFEL, but the thing to remember, this came from one single crystal, whereas the XFEL shot tens of thousands of crystals to get this structure. So there's pros and cons. Resolution is not as good, but it didn't take quite as long, and there's only one crystal versus an entire team going to the XFEL and spending a long time and a lot of sample. So there's pros and cons to both approaches. When you look at it, we could identify 4 cholesterol molecules. The drug that's bound inside of it when you crystallize it was pretty clear. So the data was really high quality. So this is exciting. So we're hoping that this is kind of the new way to look at membrane proteins, is to put this viscous membrane protein stuff on the grid and then use a cryofib to kind of get them to the ideal size. And really, honestly, for proteins, in my opinion, the cryofib is sort of going to be kind of an essential tool. I mean, unfortunately, it's a $1.5 million essential tool, but it makes the process a lot easier, because then you just find your crystals and, okay, maybe you got a great crystal, but it's like 1 micron. It's just a little bit too thick. You just shave off the top of the cryofib, put it in the microscope, and collect data from it. You don't have to worry about trying to find the thin crystals or break the crystals apart. You just use the fib mill to get them down to size. Okay, so in the remaining time here, I want to talk a little bit about small molecule work, because I know there's some people interested in small molecules, and if you're a crystallographer, you should really know how small molecule crystallography works as well. So microIDs actually can be used for a lot of samples. I mean, we initially developed it for protein work, but now we're actually doing a lot of other samples, because in the end, crystals are crystals and diffraction's diffraction. I mean, it's just how you handle things are slightly different, but in the end, the fundamentals are pretty much the same. So as long as you have a crystal and it's small enough, you can use microID on it. So we've done it on, proteins, peptides, small molecules, a range of targets. And so one of the cool things that came out, this was a paper from my old lab, and it kind of came out at the same time as another paper from Europe, where they showed that you could basically take raw synthesized powder. So you do a chemical synthesis, and you got the product, and you just take that, even though it looks like powder, doesn't look like much, but inside there is actually little tiny crystals. And then you can take that little tiny crystal, there's the pointer, and collect really high resolution diffraction data and solve your structure right from that sort of synthesized powder, without any need for going through like crystallization trials or anything like that. Now again, I want to make sure it's clear that you still need crystals. If your material is amorphous, MicroID is not magic. It doesn't suddenly turn your sample into diffracting, right? You still need crystals in your sample. I get a lot of times people saying like, oh, my crystals don't diffract. Can MicroID work? It's like, well, no. You still need crystals. It's just they're tiny. They can be tiny, but they still need to be well-ordered crystals. And so here's a couple examples of using small molecules. This first example I like to bring up because it was my first small molecule I solved at ASU, and it was kind of interesting because it actually served as a nice blind control, because we found out later after we solved the structure that the group that we work with, which is Adam in New York, they found out that they actually had solved this by X-ray like a year or two before, and they kind of forgot about it. And so we solved the structure, and then without knowing that, and then actually could then directly compare with the X-ray structure and see how closely they match. So this is this small molecule right here. If you take this sample, which is dissolved in some solvent, I forget which one this is, some sort of like dichloro something, and they put this sample right on the grid and let the solvent evaporate. And the result of that then is you get little crystals all over the grid. You shoot those little crystals like something like this one right here that I have circled, and you get this. You get diffraction that goes to about 0.6 angstroms. And then just for the crystallographers, I'll play the movie because I think it's very pretty. Your data set looks pretty awesome, just from that little tiny crystal. So we collect data from 5 or 6 of these things laying on the grid, however many we have time to collect. I think in this case we merged 3 crystal data sets together. And we were able to get the structure. And like I said, then we found out later, if you saw the structure, that, oh, we had an X-ray structure too. It was a very tiny crystal for X-ray. I think it was only like 50 microns by 50 microns or something, and that's why it took them so long. But we were able to compare, and you can see the unit cells are nearly identical. The resolution for MicroID is a little bit better, and then the crystal size is several orders of magnitude smaller for electron diffraction. But essentially, we got the exact same structure. And I can't remember how long they said it took them to get the X-ray structure, but it took them a long time to get that one crystal that was small but still big enough, whereas we did this in a couple hours. So that was kind of cool. So small molecules, you get structures that resemble X-ray crystallography. So that was a good test case. Now, what about talking about how fast can this actually be done? As I mentioned this, okay, it took a couple hours, but they had already made this sample, and they already kind of characterized the sample. They knew a little bit about it. They knew it would form crystals on the grid already. So it took a couple hours, but there was a lot of work that went ahead of that. This sample really is showing that we could get a structure in just a few hours from an unknown sample. So this is an interesting story. One of my good friends, he makes unnatural amino acids for putting them into proteins. And so they're synthesizing this unnatural amino acid. Here you see it's a tyrosine variant. They were synthesizing using an enzyme, so they thought, okay, this should work. They got the NMR results back. They got a whole bunch of powder samples. They got the NMR results back, and it was weird. It was confusing. They couldn't figure it out. It seemed like carbons were moving around. It was weird. They couldn't figure out what was going on. So my friend's complaining about this over lunch, about the arguments they're having with the NMR groups, and so I said, do you have the powder? He goes, yeah. I said, okay, after dinner, let's go to the microscope. So we go to the microscope after dinner. He takes his powder. I put a grid inside his powder, put it in the microscope. Don't even cool the microscope down. Just pop it in, and we see little tiny crystals. We shoot these little guys, and we get really high diffraction. Spend maybe 20 minutes collecting 5 data sets, and then immediately transfer the data to my computer, run it through Shell XT. We get this as the output, build the model, change the atoms, do some refinement, and we end up getting a structure. From start to finish, it was about 2 hours. And it actually showed them that, yeah, they did make something weird. The NMR people were right. The crystal was actually racemic, meaning it was being made outside of the enzyme as some weird side reaction. And what they were getting was this carbon that's supposed to be here had somehow moved to over here. So that was what was giving them this weird NMR signal that they weren't expecting. So it basically saved them 2 weeks of figuring out what is going on, because everybody's a crystallographer knows, seeing is believing, right? So okay, so they're going from NMR spectra, they're doing CD, you're doing all this stuff. Well, here, boom, here's the structure. Yeah, you've got something weird. And so that was really cool. 2 hours, that's all it took. And then there's some map. You can even see some nice density for hydrogens on there too. One more really cool thing that you can do in the small molecule world, I guess technically in the protein world too, and I'll finish up with this so there's time for questions, is looking at polymorphs. This is pretty important for pharmaceuticals because polymorphs act differently, they have different activities depending on the polymorphs, and sometimes samples will have more than one crystal form in them. Well, you can solve multiple crystal forms from the same sample using MicroID, just as long as you find those crystals in the sample. So that's what we did in this case here. We had this molecule here. It looks like this. This is from the same group from New York we worked on in the first sample. And they know that they do the same thing. They deposit on the grid, and they make thin films. This is kind of standard how they work. They deposit it, let it dry on the grid. That makes crystals. And then if they do this, what they call vapor solvent annealing, so they anneal it with another solvent and let things kind of recrystallize and reform and anneal, they get very nice, clean powder diffraction patterns right here. Very nice kind of single crystal pattern. And this has been solved by X-ray crystallography. So they know that this is what it looks like, because when you grow big crystals of this for X-ray, this is what you get. So we did the same thing. We just to test, we did it with MicroID, and sure enough, they call it the J aggregate. We get this J aggregate by MicroID, matches the X-ray structure, everything makes sense. The reason we did this, though, is because they found that if they don't do vapor solvent annealing, which for their purposes, sometimes they can't do that. But if they don't do vapor solvent annealing, they get some extra peaks in their powder diffraction pattern. And they can't quite explain it because the only crystal structure they had available is this one right here that I mentioned that we solved after vapor solvent annealing. So they're trying to figure out, well, what is this? So what we did is they make the grids the same way, deposit on the sample, let it dry, but then we don't do vapor solvent annealing. And when we go through and we look at those grids, we see some crystals that are this J aggregate. We collected actually quite a few data sets of the J aggregate in that sample, but we also found something else in there. We started seeing crystals that, by eye, you can, when you collect diffraction data, you start to pick up on these things. By eye, you say, these crystals are different than all the other ones we've been collecting. And so we started focusing on those. And sure enough, those were of this other unit cell. And you can see it's actually not very different at all. It's just these rings kind of slip and they stack on top of each other as opposed to being slightly off. And this had never been solved before by X-ray, because every time you grow a big crystal for X-ray, it always favors this other form. So you can never sort of trap this small contaminant. But MicroID, we could go through and just look at all those little crystals and sort out, okay, here's all the J aggregate, and every once in a while we get an H aggregate crystal. And when we do the predicted powder pattern for each of these, you can see that for the J aggregate, it matches the vapor solvent annealing peaks. And the H aggregate kind of explains all those little extra peaks that they saw when they don't vapor solvent anneal. So this really helped them figure out, okay, what is going on in the non-vapor solvent annealed sample. I also like to bring, so this is an important point to say, you can solve polymorphs, but it's also an important example of why when you're doing small molecule stuff, you should always try to couple it with a powder diffraction pattern. Because you can solve an entire structure from one little tiny nanocrystal. You have no idea if that one tiny nanocrystal that represents 1 to the -15th of all the sample in your bulk sample, if that represents the entire bulk. Like, for example, if we had just randomly got lucky on that H aggregate sample and caught 2 or 3 crystals of the H aggregate before we ever saw the J aggregate, we would have incorrectly said that the non-vapor solvent annealed sample is all H aggregate. But when you look at the powder diffraction pattern, you can see, okay, this powder pattern doesn't match this. There's other stuff in it. So you always want, whenever you solve a structure with MicroID of a small molecule, you always want to take a bulk measurement if you can, with powder diffraction, for example, and then do a predicted powder pattern based on your structure and make sure that you can explain the bulk sample is explained by the structure you saw from that tiny little nanocrystal. So that's very, very important. I know crystallographers, small molecule crystallographers do that as well, because you want to make sure that when you recrystallize it, it is still what's represented in the bulk. But I think people forget about it for MicroID because you're like, oh, well, it's the same sample. I just put powder on, so it is the same sample, but not always. You could get lucky and catch that one crystal that's different than all the others. So that's my my speech on why you should couple it with other methods. And the last thing I want to leave everybody with is MicroID and electron diffraction of 3D crystals is a relatively new area. So we're kind of where X-ray crystallography was, back in the 70s. We're still kind of figuring stuff out. So oftentimes you'll hear a lot of criticism like, oh, well, your R factors aren't good, or your completeness is that, well, we're still working a lot of this stuff out. So just to highlight that, I want to show you that, in 2013, the first paper we published was the first time, proteins were able to be solved from a thin 3D crystal by electron diffraction. We got about 3 angstrom diffraction. By improving the method, just a few months later, from the exact same batch of crystals, like we froze multiple grids, and so we used that same sample, and then we collected data. We improved how we collected data, and we worked with Andrew Leslie, who wrote the MOS film, if you're familiar with that program for X-ray crystallography. Just by using a better program and better data collection, those same crystals now went to 2.5 angstroms. Because the first sample, we collected data kind of in a weird way, and I was actually hand indexing the diffraction patterns. I had a ruler out, and I was measuring things, so it took 6 months to go through those data sets. This took 2 weeks, and we got better resolution. So it just highlights how the methods improvement is really driving things forward. A couple years later, now lysozyme goes to 1.8 angstroms. And now there's a paper from my old group that's in bioarchive that's going to come out soon, I think, where they coupled cryoFib milling to lysozyme, and they have 0.9 angstrom structure of lysozyme, where they could use direct methods and see all the atoms in lysozyme. So really, the methods are still, this is not a mature field. Like, X-ray crystallography isn't mature either, but, it's had time to develop, and, it's pretty good. We're still working on things. So, and there's a lot of questions I'm sure I'm going to get from the audience about that, so I'll leave it at that. So in summary, MicroID is a great way to solve structures from tiny little crystals. For proteins, it takes a lot of work. I'll say, proteins are still difficult. Proteins are always difficult, right? For small molecules, there's other difficulties, but actually it can be pretty straightforward for small molecules. And like I said, that's kind of where, even though that's not what we were thinking at the time, that's kind of where MicroID is going. Is you've got service centers going up all over the place that are just accepting samples from pharma, solving structures from, using electron diffraction. And the nice thing is, as more people use it, now it's not only 2 or 3 labs in the world that are developing it, but now a lot of people are developing it, and you'll hopefully see this method improvement I'm talking about start to really take off as more people start to work on it. So that's really exciting. You can do MicroID in a lot of commonly available instruments that many people already have, with just maybe improving the camera, for example. And now they're making dedicated electron diffraction instruments as well. So it's really an exciting time for MicroID. But don't think that just because you don't have a dedicated microscope for MicroID, you might have something somewhere nearby that could actually do it. I'm happy to answer any questions about that. Like, oh, does my microscope work? We can talk about that. And so I'll acknowledge everybody that helped me work on this, both my old postdoc lab and in my current lab, and funding that helps support this work. And with that, I'll open up to any questions.
Questions?
Yeah, okay. Thank you for this very nice talk. And I have a question. So I wonder, you have some examples of small molecule diffractions that achieve a resolution of 0.6 Armstrong.
Yeah.
I wonder how often you achieve this
Oh,
resolution.
let's see. I would say the average resolution we get for small molecules is probably somewhere between 0.8 and 0.9. I've had that sample when we finally processed it. I think we ended up taking it to 0.75. We have one that was 0.57, others that are 0.9. So the average is around 0.9, I would say. But we have to at least get to like 1.1, 1.2. Otherwise, we can't, have direct methods work. But usually, if something diffracts well, you got nice clean spots, it usually goes beyond 1 angstrom. It's the samples that are like, I don't even know if I should really be collecting on this, that you're kind of at the 1.2 range. So, but if crystal diffracts well, it usually goes pretty good, at least 1 angstrom.
Okay, and related with this, is the low temperature a key factor like in X-rays?
Yeah, so I personally haven't done like a side-by-side comparison of cryo versus no cryo, but yeah, the conventional wisdom is that the cryo helps with the radiation damage. But again, the structure I showed, the rapid one I showed that we did in 2 hours, that actually wasn't frozen. That was room temperature. So, but yeah, in theory, it should help. Just like X-rays, cryo should help, but it might not be necessary for all samples. For proteins, it's 100% necessary because you need to protect the sample from the vacuum. But I think for small molecules, it can help with radiation damage. But I've never, like I said, I've never done that comparison to say for sure. But I know other people have for X-ray.
More questions? Let's try Alman.
Thank you very much. Very nice talk.
Thank you.
I've got 2 questions. First one is about the unit cell. What is the largest ever unit cell used for cryo?
I think it is the catalase one, which is like it says 70 by 100 by 200. I think.
So it's not expected to have crystals to collect data from crystals with larger.
I think you can. I mean, if they diffract,
overlap.
So there's tricks you can do. Like I've never done it because we never had one. But you can do tricks that equivalent to like swinging the detector out. You can make your camera length long to get them to spread out, and then you can maybe move the beam to the corner of the detector. Okay. So you can do tricks. I've never done them, but they should work. Rather than because we don't move our detector, it's all virtual. So rather than swinging the detector this way, you can just move the beam.
And the other question is, I've got the impression that the electron density maps look different than the ones you see by conventional X-ray crystallography. I mean, they are like the blobs are different. Why is this?
They're mostly similar. So they're with X-rays they're electron density maps, and EM they're Coulombic potential maps. But for the most part, they look the same, but there are some differences around, especially around charged residues, because electrons interact with charge, whereas X-rays don't. And so you will see some of that.
So there is a physical reason.
Yes. Yeah, and that's actually one of the reasons that, that's my hypothesis. One of the reasons why our R factors are higher is because all of our scattering factors, our electron scattering, we use electron scattering factors, but they're all derived from X-ray scattering factors. And that's all still assuming spherical atom and isolated. These atoms are isolated. Well, in these structures, the effects of chemical bonding and of charge are real, and electrons see them, especially at low resolution. All that effect is at low resolution. But we're not modeling any of that. We're still assuming spherical, isolated atoms in our scattering factors. So that's why when I say, I don't necessarily think it's our F observed that's giving us the high R factors. I think it might be our F calc isn't being modeled as good as it could be.
Jordi?
Yeah, also thanks for the nice talk.
Thank you.
I have a question related to small molecules and proteins. So you said something at the beginning about energies, but when you think about X-rays and you want to measure small molecules, normally you go to a short wavelength, and if you go to proteins, you go to a short, let's say a longer wavelength, so that you get a better spreading of the peaks or the signals on the detector, no? And now we are thinking to get a MicroID system which has a fixed wavelength, no, of 200 kiloelectron volts, and this means it is a wavelength of 0.253.
Yeah, I forget what it is. Yeah, something like that, 0.2 something.
And so our first intention was to measure small molecules, and it seems to work, no, with this wavelength. But of course, we got now several questions about people intending to measure also proteins. And do you think this wavelength would be okay for measuring in good conditions?
Yeah. So I've collected data at both 200 kV and 300 kV, both proteins and small molecules, and they all work fine. So the original work I showed when I was in my postdoc, that was on a 200 kV instrument. And I know they've done then small molecules on a similar instrument. At ASU, we have a 300 kV instrument. And so all the work I showed later, that was all for protein, and that was all on the 300 kV instrument. And so while there are differences between 200 and 300 kV, and you could make an argument for which one's better. I haven't seen like, oh, this one's better for small molecules, this one's better for proteins.
Okay, and then just a comment in relation of the seeing the reflections different. I mean, one of the important points are the dynamical effects
Right.
on the diffraction, because this makes a very strange view. So you have very intense peaks and weaker peaks.
Right. Yeah. So multiple scattering is what actually people thought, in a long time ago, that you'd never get this to work as a multiple scattering, which is true for like really perfect crystals in material science. That when people were making these theories, we found that because proteins and small molecule crystals aren't so perfect, it actually helps us because we get less multiple scattering. So we see that multiple scattering roughly is like maybe 2 to 5% error that we get from multiple scattering. And it's mostly because we're collecting data continuously. We never really hit the zone axes. That's where dynamic scattering is the worst, is when you hit a zone axis. And very rarely do we actually hit one of those. So yeah, but that's another thing. If you then do dynamical refinement, you can see your R factors can drop by a few percent. Nothing like crazy. It doesn't go from like 20 to 1. It goes from like 20 to like 15 or 20 to 40, but it's there. And the nice thing about dynamical scattering is it's kind of the equivalent of anomalous dispersion, so you can use it to phase, or use it to determine absolute structure.
Okay.
I think that's the important part of dynamic scattering, not the R factors. Like, okay, a few percent R factors, that's fine, but like being able to phase or absolute structure, so it actually becomes a feature. That's why I always thought it would be. That's why I always think it's cool, like what Lucas Platanis is doing, because it's basically taking the thing that everybody said, oh, electron diffraction can't work because of dynamic scattering, and it's actually turning it from a weakness into a strength.
Yeah.
So I think that's kind of cool.
Okay, and I agree totally with you, and it's very interesting for us because of absolute
Right.
configuration and pharmaceutical compounds. I have a last question related to what Eduardo said in relation to the temperature of measuring. So, in fact, when you measure X-rays, besides that you need to sometimes to protect your sample and measuring cool, it's also the fact that when you measure lower temperatures with X-rays, normally you get better data, no? Because you get a better,
You get it's a bit, yeah.
And do you have any idea if also a part of the destruction of the crystal, if you see some improvements in depending on the temperature, or
Like I said, I haven't, and the main reason we haven't done it is because I don't have a microscope right now that we can sort of do like room temperature and then cold. So like all of our measurements that have been at room temperature have been on a different microscope so it's hard to say the differences. It's like changing 3 parameters at once. But I think everybody right now is just assuming like, oh, all the work that's been done on X-ray crystallography is going to hold for electrons. But I think eventually somebody will probably do that experiment. Maybe it'll be us. I don't know. Maybe it won't be. But I think someone eventually is going to do that experiment. Like, here's a crystal, room temperature. Here's a crystal cryo. What's the difference? My guess is it's going to be similar to X-ray.
Okay, thank you very much.
Yep.
Josemi.
Thank you very much for your excellent talk. I would like to know some comment from you about the tilting range with low symmetry crystals.
Yeah.
Did you find any limitations with this?
Yes.
these
So
crystals?
this happens a lot with, especially small molecules, because small molecules are mostly like 90% of our small molecules end up being P2. And I from what I've heard, I'm new to small molecule, but what I've heard is that's not uncommon. That most small molecules are P1 or P2. And very rarely do they get big. Yeah. Yeah. So there it becomes kind of a problem. So that's where merging multiple crystals comes in, is you hope they kind of randomly orient and you get different views. If they don't, like if you have a plate-like crystal that all lands in the same orientation, you're going to be limited to probably 80% completeness, which in our experience still works. You can still get structures from 80% completeness. But there's tricks you can do. You can potentially like if you use carbon, like holy carbon, you can actually like kind of make the carbon a little broken. So you get like a little flap of carbon that's dangling a little bit. And then now that preferred orientation is still preferred orientation, but the grid is moving down a little bit. So there's kind of tricks you can play, but at the end of the day, sometimes you're just limited by, the geometry of the crystal. It doesn't want to lay in different orientations, and then the symmetry. For proteins, it's a little bit less because a lot of proteins are more symmetric than small molecules are. But still, usually for proteins, you have to hit 1 or 2 crystals. Catalase, for example, those were flat plates. We only got to 80% completeness. There's no way we could get more because it was a flat plate and every crystal laid so the C-axis was parallel to the electron beam. And there's nothing we could do about that. So that's what's known in the 2D crystallography world as the missing cone problem, where you just have a missing cone of reflections. But like I said, we found that if you can get up to 80-85%, it's not such a huge problem.
And about the nature of submicrocrystals in your hands, do you think they are as homogeneous as larger crystals, or did you find some more heterogeneity?
Yeah, so that's an interesting question. So because I know, like I heard Jose might be able to comment on this too. Like, one of the arguments for Excel is like, oh, microcrystals would be better ordered than big crystals because you get little domains. I don't know if that's really been shown to actually be true. We get a big crystal that diffracts at 2 angstroms. The small crystals will diffract 2 angstroms too. What we have noticed, which is really cool, is one of my colleagues, they did 4D STEM on a microcrystal, and they found that even on a microcrystal, these are peptide crystals that diffract to 1 angstrom, that actually it's a lot more mosaic than you would actually think. That like when you're diffracting from this 1 micron long by say half a micron wide crystal, that actually you're only diffracting for small little bits as it's rotating. Like this domain will light up, and then this domain will light up, and like it's not all one crystal, which is actually what helps with the multiple scattering. Is you're never actually, while the crystal might be 100 nanometers thick, you might only be diffracting off of a 20 nanometer thick little domain within that section. So yeah, in my, again, I don't focus on this, but in what I've read and what I've seen, the microcrystals are actually a lot more disordered than we would probably think they are, but they still work. So
Okay, thank you very much.
Okay, there are a couple of more questions from the people attending the seminar online. So I'm gonna go ahead and read them for you, okay? So first question from Douglas. He says, great talk. Is it possible that some of the electrons are captured by the sample? Could perhaps slow electrons be used to reduce a sample, like aromatic system, and study the reduced states? Thank you.
yeah, so actually, I mean that's kind of what a lot of the radiation damage process is in the electrons. Is they're coming in and the sample's absorbing the electrons. This whole idea of what does that do to a sample? I think people, especially material science, think about this a lot. I don't know enough to really comment on like specifically aromatic systems, but for example, that's one of the reasons why we're mentioning the charge stuff. Sometimes you don't see the charge groups as well because, like I said, electrons they don't interact with the charge as much, but also because those get damaged faster. And that's been seen in single particle cryo-EM, not just diffraction, that the charge groups either repel the electrons and they kind of stay around a lot longer, or they attract the electrons and those things get damaged faster. I don't know if I answered the question, but the electrons do get absorbed by the sample for sure. What does that mean, and can you use it? Maybe if you're smarter than I am with that, you can. Yeah, and that is the point. It would have to interact with the crystal, but not kill it. Which there might be a window in there where it's interacting, but you're still getting diffraction. But yeah, it's an interesting question. I have to think about it a little longer.
Okay. So second question from Miguel Daniel. He says, great talk. It was really interesting. I have couple of questions. First one, does making crystals thinner not damage them? And if not, then what is the limit? Second question, will thinner crystals always yield better resolution?
Okay, good question. So thinner crystals, better resolution. Yes, as long as you have enough layers to give you good diffraction. So if you have a single layer like 2D crystals, you actually don't have Bragg spots, you have Bragg rods. And so you need enough layers to kind of collapse those down into like actually Bragg spots. But once you have enough layers to get good diffraction, then yeah, thinner is always better. So there's some optimum where maybe going from 5 to 6 layers gives you more unit cells, better diffraction. Maybe going from 6 to 7, now you're just getting more absorption through that thicker sample. So there's going to be some optimum, and it depends on the unit cell thickness and all that sort of thing. In terms of cryofib milling, there is a thin layer that gets damaged by the beam. It hasn't been well characterized yet, but that's what, for example, when we make the lamella, we'll cut it to say like 300 nanometers thick. Probably there's like say 50 nanometers of damage on the top and the bottom, so you really only have the middle that's diffracting well. But I mean, the part that's not damaged diffracts really well. Like I mentioned before, that paper that's going to come out soon from my old lab, we have 0.9 angstroms from lysozyme. I mean, if that crystal was damaged, you wouldn't get 0.9 angstrom diffraction.
Okay, thank you, Brent. No more questions from, Is there any other question from the audience here? If not, I think we can maybe, we can wrap up the.
Important is that as invited speaker, please, this is a small present for you.
Oh,
This one.
thank you.
This is a sign of the Institute for all invited speakers. So thank you very much.
Oh, thank you so much. Very nice. Thank you. Thank you, everybody.